Oops, sorry. All right, let's get started. I didn't even know it was supposed to rain today. None of you look very wet though, so that's good. So we are still a little more than a week out from our next exam. So next Friday is our next exam. I have it sort of in my mind what I'm hoping to get through and what I'm hoping that will cover, but let me wait till Friday. Hopefully I can pin that down a little bit better on Friday, and I'll announce starting back with chapter 15, which was just some a lot of terms, epidemiology terms, innate immunity, adaptive immunity. We're going to finish that today and start vaccines. Probably next time we will get through the staphylococcus. So I'm trying to decide whether to include the streptococcus on the next exam as well as the staph. So more about that. There are a few questions on staphylococcus in section 20.2. Again, I'll make some announcement about that. There's only a few questions there. I hope to prepare more in the future, but haven't yet. So there's only a few questions on that. I just made that available like a day or two ago. So, okay, so that's kind of what looking forward. Let's look back a minute and have a couple of practice questions about what we covered last time. Okay, so take a minute to read this one. If you haven't yet, I'll give you 20 or 30 seconds to do that. Make a choice. Okay, so remember there's two different ways that B cells can become activated. When they become activated, they're going to differentiate into plasma cells and make antibody. So those two ways, it depends on what kind of antigen is acting on the B cells, or that the B cells are recognizing with their B cell receptors. All proteins are going to cause a T-dependent activation, meaning they need some stimulation from T helper cells that are also activated. But which of the following characterizes a T-dependent activation of B cells? Okay, so these first two are going to be T-independent. Remember, that's when T helper cells are not necessary to activate the B cells. Okay, that makes a fast response, but usually not quite as good of one, because there's no memory cells and you only get one kind of antibody. You don't get any class switching. Okay, this last thing here, I just made that up. The correct answer here is a subset of T helper cells are going to secrete cytokines to stimulate B cells to make antibody. Okay, I want to back up just a second to that slide where I talked about T-independent antigens. And again, don't need to go back to that if you don't want to. But I remembered when I talked about those T-independent antigens that I had added this little bullet point here because they talk about that in your textbook. And that's a detail, I forgot to even mention it in class because I haven't really taught that before. But there's certain kind of antigens, right? We said mostly these polysaccharide antigens, usually associated with the bacterial capsule, that can function to activate B cells directly. It turns out there is some second signaling that's involved there. Apparently, B cells also have toll-like receptors. Remember, that's a kind of receptor that we talked about is found on like phagocytic cells like macrophages and neutrophils. That the toll-like receptors recognize broad-based pathogens, PAMPs, pathogen-associated molecular patterns. Okay, so it turns out there is a second kind of signal that's involved there with those T-independent antigens. I would say don't really worry about that. With any textbook, they decide how much to simplify, how in great detail to go into. I just wanted to go back and say I forgot to even mention that. I blew right through it in class, so don't worry about it. That's what I'm trying to say here. Okay, so again, T-independent antigens, fast, usually bacterial capsules. A T-dependent antigen then, in order for the B cell to get activated, it needs to have an activated T helper cell that's going to secrete cytokines in order to do that. Okay, so we'll touch back on that again. Let me ask you another question. When we ended last time, we were talking about what goes on in our lymphatic system. So let me ask you a question about that before I finish up talking about that. So give you a second to read that. Okay, so some of these things we talked about last time, and I think, I got to thinking about maybe like the main point, though I didn't really hammer home. So which of the following is false? Okay, concerning lymphatic system. First one's true. Yes, lymph moves through lymphatic vessels really slowly because that's just smooth muscle contraction that moves that. Okay, it's not pumped. We don't have a lymph pump. Yes, T cells are made in the red bone marrow and then they travel to the thymus for maturation. Yes, valves ensure the lymph moves only in one direction. This is the one that's false. Okay, most B and T cell interactions with antigen presenting cells occur in the thymus. How could we make that true? Nope, they're not that. Those interactions are going to happen in the lymph nodes. Okay, and then this last one is true. Sorry, is true also. Lymph drain from the tissues into the lymphatic capillaries and then travels typically to a lymph node. Okay, so where we ended last time, we were kind of a close-up of a lymph node. I didn't even label that very well. Okay, so remember we have somewhere between 500 and 700 lymph nodes throughout our body. They are concentrated in 3 areas of their body. We have a lot of lymph nodes in the groin area, in the armpit area, and in the neck area. So lymph travels to these lymph nodes. There are more afferent lymphatic vesicles or vessels, meaning vessels leading to the lymph node, and usually much fewer leaving the efferent lymphatic vessels. That's so that lymph has a chance to mix around. So remember what's traveling in the lymph. We've got lymphocytes, macrophages that are presenting antigen. Hopefully will come in contact then with the very few B cells or T cells that we have that are specific for that same antigen. And also there's blood supply here too. So there's lymphocytes in the bloodstream that can be mixing with cells that are in the lymph. Typically, if a patient is experiencing an infection, they are going to have, more cells, more white blood cells proliferating, more fluid, more inflammation. So the lymph nodes can enlarge during an infection. And that's one way, often if you go to the doctor will check and see if your lymph nodes are enlarged, maybe around your neck area. And that's just, again, another sign that our body's fighting off some kind of infection. Once the infection is tamped down, the lymph nodes will shrink back down again. Okay, so we are nearing the end of our discussion here. We need to talk a little bit more specifically about T cells. I mentioned a few of them already, we talked about them briefly already. So we're going to talk a little bit more specifically and then talk about how T cells get activated and then what they actually do when they're activated. All right, so cytotoxic T cells, 3 different kinds of T cells. We mentioned the cytotoxic T cells, the helper T cells. There's actually a 3rd type of T cell that I didn't mention before called regulatory T cells or Tregs. That's kind of, I'll just mention them here, but it's kind of beyond our scope of discussion. They do help to repress the adaptive immune system. We're not going to go into the mechanism of how they do that. I'll just mention them just for sort of the sake of completion. Let's fill in a few more details about cytotoxic T cells. We've mentioned that their job is to kill cells, kill our cells, kill host cells that are infected with some intracellular pathogen. So an example of that would be a virus. They are going to have a little receptor on them called CD8. CD stands for cluster of differentiation. Okay, these are glycoprotein receptors. There's many different glycoproteins like this CD. There's like CD, lots of different CD numbers on the outside of immune cells with various different functions. We are going to really, they help us identify what kind of cell that is. So if we find a cell that has this CD8 on its surface, then we know that's a cytotoxic T cell. Okay, the cytotoxic T cells, we've mentioned this already, but they are going to recognize antigen presented by MHC class 1. Okay, so remember those MHC class 1 is what's going to advertise on the surface of infected cells what's going on inside the cell. Okay, we said that helper T cells are sort of the general in charge of the army. They're coordinating lots of aspects of the immune response. They have a different glycoprotein on the surface. They have CD4 glycoproteins. Okay, and they are going to recognize antigen presented by MHC class 2. Okay, and we mentioned this kind of briefly before when we were talking about B cell activation, but there's actually 2 different subsets of T helper cells. And we're going to go through this in a minute, but once the T helper cells are activated, they are going to differentiate into either T helper type 1 or T helper 2 cells. T helper type 1s are the ones that are going to activate the cytotoxic T cells, and then T helper 2 are going to activate the B cells. Okay. All right. So one more thing to mention that I don't think I mentioned before, we're going to look at the T cell receptor. And I just put it, remember we talked about the B cell receptor already. There are some similarities of T cell receptors and B cell receptors. Let me just write that in here. They both have an antigen binding site. Okay, so the T cell receptor looks a little different. With the B cell, it has 2 antigen binding sites where, again, the T cell looks a little bit different. And then they both have a constant region that's going to be embedded in the plasma membrane of the T cell. Okay, so B cell also has constant regions that's embedded in the B cell membrane. Okay, so there's going to be thousands of these in for every T cell. Okay, so again, those are embedded in the membrane all around the T cell. Same thing with B cells. They've got thousands of them. We draw one, but there's a ton of them there. And they all have the same specificity. In terms of each cell, like all the T cell receptors on one cell have the same specificity. So that's kind of, again, very similar with B cell receptors. What's different about T cell receptors is that they're very particular in how they notice antigen or how it's presented to them. They are going to only recognize antigen that's presented with MHC class by MHC antigen. Oh, there it goes. Okay. Oh, sorry, I'm reaching for my notes here. So T cells only recognize antigen give myself very much room here when presented by MHC. Okay, so again, they're kind of particular. Remember, B cells, they can recognize antigen directly. Okay, so those B cell receptors will be specific for some epitope of antigen, where the T cells, it has to be presented to them by MHC. Okay. Whoops. Need to do that. Whoops. Okay, so let's get into kind of the nitty gritty of how T cells get activated. Okay, so usually it's going to require T helper cell activation first. Actually, my understanding was that the cytotoxic T cells always had to be stimulated by T helper cells before they would act. Your book is a little bit cagier on that. That says, well, it's a much better response if the T helper cells are activated as well. So it's kind of hard to speak in all or none terms with lots of things in biology, and this is no exception. Okay, so first, but let's look at how T helper cells get activated. So here you have some naive helper T cell, some antigen presenting cell over here, could be a macrophage, could be a dendritic cell, has phagocytized some antigen, and then the parts that it has chewed up, it presents small epitopes from what it's chewed up by MHC class 2. Okay, so here's a close-up of that synapse there. So here's a helper T cell then that has that CD4 glycoprotein. That the CD4 actually binds to the MHC class 2, kind of stabilize that immunological synapse. This little red dot here, I'll color it blue. That's the antigen that it's presenting. So some T helper cell that has a T cell receptor that's going to fit exactly with that antigen will recognize it. Okay, so once that synapse has been made, and again, this is going to be happening in the lymph, where probably in a lymph node where lots of cells are mixing together. They find each other, might take some time. But then that dendritic cell will start secreting cytokines and we're not going to talk about which ones, we'll just call it certain kinds of cytokines that cause that helper T cell to differentiate. And apparently it can differentiate into 3 different kinds of T helper cells. Either a T helper 1 cell. Okay, and remember, these are the ones that will act on cytotoxic T cells. The T helper 2 cell subset are the ones that act on B cells to activate them. And then apparently you get some memory helper T cells, which will speed up the response the next time the host encounters that pathogen. So that would be in a secondary immune response. Okay, so that helper T cell doesn't need to find that antigen. It doesn't need to take so long. It's going to have a much quicker response. Okay, so put a pin in that. That's just kind of the activation of the T helper cell. Then if we look at the cytotoxic T cell, okay, remember cytotoxic T cells are going to recognize antigens that's presented with MHC class 1. So you have a host cell that's been infected. And remember, it's an MHC class 1 that advertises what's going on inside of that host cell. So a little bit of that virus, for example, gets presented by MHC class 1. It's the cytotoxic T cells then with their CD8 cluster of differentiation that binds that MHC class 1. And then at the same time, hopefully you're getting cytokines from that activated T helper cell. It's actually a T helper 1 cell that has been activated by the same antigen. Whoops, we'll label this as cytokines. That if that a T helper cell has been activated as well, this is going to be a much longer lasting response. Again, apparently that's not absolutely necessary, but it makes it a better response. Once that cytotoxic T cell has been activated, now it's going to start secreting a couple of proteins. I think they're considered cytokines as well. We'll call them proteins that act on this infected host cell. Okay, I like this picture a little bit better of what that cytotoxic T cell is actually doing. So here's the cytotoxic T cell here. Once it's been activated, it's going to start secreting these 2 proteins, perforin and granzyme, on the cell that's been infected, the host cell that's been infected. What do those do? Perforin kind of does what it sounds like. It perforates or pokes holes in the host cell membrane. And that allows the granzyme, the second protein, to enter. And we won't go through the mechanism of how this works, but that induces the host cell. It's like a programmed cell death or host cell apoptosis. Kind of induces cell suicide, basically, which is really the best best way. You don't want that host cell to lyse, because maybe that's going to be a means to release a lot more virion particles if this is a virus. So it just induces the host cell to die. Okay, I noticed a few of you have gone ahead, kind of gotten ahead of me in lecture in terms of answering the homework questions. I think we're in section 17.4 with this material. There's a question about this in the homework that I had to read it like 4 times before there's 2 answers that seemed like it was saying almost the same thing. So read that question really carefully that involves perforin and granzyme. It's tripped some of you up that have kind of worked ahead. So question, Ben. So question is, can cytotoxic T cells also bind to MHC class 2? I believe no. They only recognize antigen when it's been presented by MHC class 1, because it's that CD8 that recognizes only the MHC class 1. Okay, good question. Let's see, what did I forget to say? Questions here. So we want to make sure there's lots of checks and balances in lots of different immune reactions, because we don't want our immune system, especially with a cell-mediated response like this, we don't want our cytotoxic T cells killing one of our host cells unless it's absolutely necessary. So it's good to have kind of lots of things that have to be satisfied before it will do that. Same thing, we don't want our B cell making antibodies to things that are self. Okay, we want kind of lots of check all the boxes before it does that to make sure it's an appropriate immune response. All right, so one last thing to kind of circle back to as we finish up here. We talked about the different kinds of antigen, exogenous, endogenous, autoantigens. Remember, autoantigens shouldn't elicit an immune response as long as everything is working the way it should be. We talked about superantigens. So, and we said that superantigens are going to cause an unusually large number of T cells to be activated. So now we maybe understand a little bit more about that. So here, in figure A here, this is just showing a typical antigen, I believe. So I don't know, we'll write it here, typical, where you've got a macrophage presenting some chewed up antigen by MHC class 2. Here's a T cell, and it's the wrong shape here. So that epitope's not recognized, that T helper cell doesn't get activated. That's the way it's typically will work. We have many, like, a huge repertoire of T cell receptors. Their specificity gets generated the same way that we talked about the B cell receptors, how it's a recombination event that pull off that VDJ recombination. Okay, T cell receptors, same way. So we have tons and tons of T cell receptors, only a few that might recognize a specific antigen. But if the antigen is a super antigen, then there is some mechanism. So many T cells get activated. And even though it doesn't quite recognize that exactly. So these little brown spots, that's showing lots of cytokines released, way more than what is appropriate. And so the host will actually experience such overwhelming inflammation that the patient can go into shock. Okay, so blood vessels get too leaky, too many cells, too many fluids leaving the blood vessels, and the blood pressure gets really low and organs start to shut down. Okay, I like this picture. I didn't post this, but this is just showing another way to see the same thing. I like this the way it actually shows, we have many, like 20% of a host T cells can get activated. Again, lots more cytokines and lots of inflammation that will result from that. So again, that's just showing basically the same thing. But, okay. Questions before we move on? All right. Well, actually, your book it's a good segue into talking about vaccines. We need to understand some of the immunology before we kind of understand how vaccines work. We like to use terms, specialized terms, to describe just about everything, in case you haven't noticed. So before we talk about some specific vaccines, we need to kind of understand how an individual can acquire immunity to a disease. Okay, so there's different ways to describe that, whether that acquired immunity, and what I'm talking about is an adaptive immunity, a specific immunity, whether that immunity is artificial or natural or active or passive. Okay, so if that immunity is artificial versus natural, basically the way I think about it is, if it's artificial, it involves some kind of technology, and a natural immunity does not involve technology. So for instance, I think of, well, if it's artificial, could we do it 500 years ago? No. Okay, then okay, it'd be natural if we could do it 500 years ago. If we could only do it today due to some technology, then it's artificial. Sorry, did I say that the wrong way? Okay, I think you get what I mean. Like natural if it didn't involve technology. Okay. Immunity is active if the patient is making their own antibodies and it's considered passive if the patient is just receiving antibodies that are preformed from someone else. Okay, so let's put those terms together and think up some examples. Okay, if somebody has naturally acquired active immunity, that means they acquired, an example of that is when a patient gets the disease. Okay, and then, so disease, and then is immune to it because they've had it once. Okay, so I had the chickenpox when I was 8. That's an infection that once you have it, we have a really good memory B cell, memory T cell response that persists usually throughout somebody's life. Okay, so typically you get that infection once and then you're immune to it to the rest of your life. Not all infections have such good immunity. Probably a number of you have had COVID a couple of different times. But that, and chickenpox doesn't change quite like COVID has. Artificially acquired active immunity. An example there is when somebody gets a vaccination. Okay, lots of different examples. If you get the vaccination, that's basically your primary exposure. You're teaching your immune system to recognize that pathogen. You've got good, hopefully, B and T cell memory. Sometimes we don't get both, but usually, even if we just get B cell memory cells, then that's enough. And so that the second time you're exposed to that pathogen, your memory cells will conquer it even before you know that you're sick. Naturally acquired passive immunity. I think the only examples of that are mother to child, okay? Either across the placenta or breastfeeding. Okay, so that's what the picture is there. Okay, so it's natural, right? It doesn't involve any kind of technology. And the mother is just passively sharing her antibodies, either, again, through the placenta or through her breast milk. Okay, those antibodies are going to last maybe for a time, and then they'll degrade. So the baby's not making his or her own antibodies. And then artificially acquired passive immunity. That's what this last picture is down here. A patient just receiving an antibody infusion. Okay, so we sometimes, there are people that have maybe a genetic disease where they can't make certain kinds of antibodies. So those people can go in every 4 or 5 months and get antibody infusions to protect them from infections. We've seen people getting antibody infusions to help them help treat COVID. We tried that with Ebola, people that had recovered from the disease, and then harvesting those from people that have recovered. Okay, so basically there's some examples of 4 different ways that we acquire immunity to an infection. I had an interesting conversation with a friend about a year ago about the COVID vaccine. And I will admit that I am a little bit more at peace about whether people get vaccinated or not to COVID. But at that time, the vaccine was new, and this friend had said, well, that she refused to get vaccinated. And her reasoning was that she said, eventually everybody will just have COVID antibodies. And I kind of took a deep breath, and I didn't say this at the time, but I thought it later. These are the 4 ways that we get antibodies. Either you get the disease and then your body makes them. Either you get vaccinated, or you get an antibody infusion, or again, mother to child. So assuming this friend was hoping not to get the disease, then she may have to find a lactating person that had recovered from COVID in order to get those antibodies, which is not very likely. So, we don't just breathe in antibodies from the air, right? We get them discrete ways. So, and this friend admitted that she would rather get COVID than get the vaccine. And, I'm not for mandating people get vaccinated, but if you do get any disease like COVID, you have the potential to pass it on to other people. So that's something to think about if you haven't already. Question, comment? Great question. Once you have antibodies, you have them forever. So we think that the memory cells persist for a long time. And there are some antibodies that seem to persist for months. We said the IgG antibodies are kind of longer acting. They stay around for months and months. Probably not a lifetime. There is evidence that for some infections, you might make antibodies at a really low level for a long time, but it kind of depends on the infection. At least with COVID, because that virus is changing over time, then eventually those antibodies aren't going to do you as much good as what they originally did. So I think there's a lot that we don't know about that yet. Another question.
So when you receive a vaccination, you also create memory cells, do have the same strength of the immune response, because if you only have one specific antigen,
yeah, so great comment. Once you get a vaccine, is a vaccine as good as getting the infection? Usually not. So we'll talk about that. That's a great question. And isn't your antibody response only going to be to certain epitopes, whatever was presented in the vaccine? And that's true as well. Yes. So that's a good segue, your question, to our discussion of vaccines here. A couple more introductory remarks. I'd like to show this picture just to show that there has always been some resistance to using vaccines. Now, this picture, I believe, was drawn in about 1803 by the Anti-Vaccination Society. Of course, at that time, 1796 is when Edward Jenner, we give him credit for coming up with the first vaccine, and that was to smallpox. He noticed that milkmaids that contracted cowpox very rarely, if ever, got smallpox. So he said, hey, let's expose people to cowpox. We'll take some lesions from somebody that's having cowpox, rub them into somebody else's skin, and then that turned out to have a very protective effect against getting smallpox. Here's what cowpox looks like. Like, man, no thanks. I don't want that either. And we are going to talk a little bit more about smallpox in relation to monkeypox in a few weeks. But there's Edward Jenner on the left there, and that's what cowpox looks like nasty, but apparently not something that's going to kill you. It obviously also, like in this picture, you have, somebody throwing up a cow or giving birth to a cow. Obviously it didn't do those things either, but people were afraid of things that they didn't understand how they worked, and that's still today. Like when we don't understand, we're a lot more likely to be fearful of it. Okay, so some terms: vaccination versus immunization. To most people, those mean the same thing. Okay, vaccine is when you get a shot. Immunization is what happens in your body. You make B cells, memory B cells, and memory T cells with that specific, that will respond to that specific epitope. Give Aaron Rodgers some credit. He knew there was a difference between those two things. And about a year ago, when he claimed he had been immunized, well, in most people's minds, oh yeah, he got the COVID shot. No, he claimed he'd been immunized because he took some herbal supplements that he said would boost his immunity. Okay, no herbal supplement is going to give you memory B cells and T cells to a specific infection. So yes, he did get COVID. So anyway, they're closely related things. Usually people, it's really very much the same thing. And he used it in a different way that wasn't quite straight. All right, so why do we want to vaccinate? Like COVID aside, what's the good of vaccinating? We typically want to especially vaccinate young children and babies because their immune systems are just not quite as mature. Okay, so they're going to have an immature immune response. Okay, we talked about that, especially with those T-independent antigens, how young children don't have a very robust response. Their B cells just aren't mature enough. So we especially, and we talked about how a while ago that in 1900, about 1 in 5 children didn't make it to their first birthday. If you go back to 1800, it was about half children didn't make it to their 5th birthday. So just the so many vaccines that we give children now, that's greatly influenced improved life expectancy and protected children. We also want to protect older adults because as we age, our immunity wanes. Okay, so ask your grandparents at Thanksgiving what vaccines they've had lately. Hopefully they're keeping up with that. The point is, with vaccines, we want to get to herd immunity. And that just means that if we get a certain percentage of people that are immune to an infection, there'll be so few people that are susceptible that they're not likely to come in contact with each other, and therefore the infection won't circulate anymore. So, what do we need to get to herd immunity? It depends on the infection. Usually somewhere between 70 to 95% vaccination rate. With measles, it's like 95 because that infection is so contagious. I just read recently with the flu, they think it's more like 30 to 40% because, you know, even though it's contagious, it's just people are way less likely to get it. If even we could get, you know, 30 to 40% of us vaccinated or immune. Okay, so the idea is that if most people are vaccinated or not susceptible to an infection, then the people that can't be vaccinated will be protected. So why can't people be vaccinated? Well, young children or young babies. There's some in vaccinations that aren't given till measles, for example. We don't give that to babies till they're 12 months old. Why not? Well, we could give it earlier than that, but it doesn't produce the memory cells that are needed because they just have immature immune system. So we have to wait till 12 months so it'll do any good. So babies are susceptible to measles until they get that first vaccination. So we want to protect babies. There are people that are immunocompromised, that even if they got the vaccination, it wouldn't do any good. Their adaptive immunity doesn't respond very well. And there are people that have allergies to vaccines. Okay, so those are the populations of people that really can't be vaccinated. Okay, so if the rest of us that can be get our vaccines, and we can protect those vulnerable populations. So I think that's really the best argument to you know get our vaccinations whenever we can. Okay. The goal of vaccination, of course, is to induce adaptive immunity. Okay. We want to get memory, ideally both memory B and T cells. Sometimes the T cell memory cells are a little bit harder to come by, and we'll talk about a little bit why that is. Okay. All right, so okay, so let's start looking at the types of vaccines in use now. We'll continue this next time. The first vaccines that we had were live attenuated. And that first smallpox vaccine, it wasn't even attenuated. Attenuated means weakened. It was full-fledged cowpox, and again, that was kind of a weakened form, or it was close enough to give immunity to smallpox. But with these vaccine, the agents have been changed in some way so that they're not going to cause disease, or at least not cause full-blown disease. How do we weaken them? Sometimes we'll grow them in a cell type that's not its normal host. So for example, for the rabies vaccine, we grow rabies virus. Instead of growing it in human nerve cells, which is its normal target cell, we grow it in monkey kidney cells. So apparently that induces changes in the virus that make it no longer able to cause an infection. Sometimes we might grow the pathogen at a different temperature, or in the case of the tuberculosis vaccine, it's been cultured for so many generations that it's a strain that's lost its ability to be infected. All right, so big advantage with this kind of vaccine is that it grows and multiplies in the host, very similar to a natural infection, and we get a more natural adaptive response. It's infecting cells, so the cells are presenting antigen with MHC class 1, and we're getting good T cell memory as well as B cell memory. So typically you only have to give maybe 1 or 2 doses of this to give somebody long-acting immunity. And you could think of this as an advantage, is that if you have a population maybe that's very remote in a jungle or whatever, if you can get some of the population vaccinated and it causes a mild illness, they might pass that on to other people around them that weren't vaccinated, and thereby spreading immunity that way, even though not everybody was vaccinated. So we call that contact immunity. That can also be a disadvantage of an attenuated vaccine. We'll say we don't always want contact immunity. Oops. At least in this country, we don't want to pass even a mild form of an infection on to any vulnerable population. You wouldn't want to accidentally get a baby infected, for example, or an immunocompromised person. Okay, we will pick up there next time on Friday. We'll finish vaccines and start Staphylococcus. All right, have a good day.